Hello, this is the fourth class for using R to handle laboratory data. Today, we're going to be talking about something very big. We're talking about Bioconductor. All right, so Bioconductor is a range of R packages designed for analyzing biological data. The Bioconductor project aims to develop and share open source software to precise and repeatable analysis of biological data. We foster an inclusive and collaborative community and develop developers and data scientists. All right. So this is a big project that is can be very useful if you find the right tools in it. So first off, we're going to go to the Bioconductor page right here, and it's bioconductor.org, and it has a bunch of information. They have books and learning and a bunch of resources about how to use Bioconductor. All right. Secondly, we're going to look at how to install Bioconductor. It's pretty straightforward. It's just a package essentially. You just put these in and run this code, which I'm about to do myself actually right now. Run it. All right, and then so you run these two pieces of code. Make sure that it's you have the up-to-date version of R, because I originally when I tried to update this a while ago, the it wouldn't work, and it's because I didn't have the updated version of R. So Bioconductor is fairly usually uses the most current version of R, so make sure you update R. All right, and that. And sometimes they ask, hey, do you want to update things? Usually just say no. Like A is all, S is some, and N is no, just say no. But sometimes if the Bioconductor doesn't work, it's because something's not updated. So just let it update everything. But for purposes today, I don't think I need to because I've recently updated my programs. And then downloading these two core packages for Bioconductor. And once again, it's going to ask you, hey, do you want to update everything? No, you don't have to. If your things are up to date, that's how you install Bioconductor. But now let's actually get into Bioconductor packages, which is what's actually interesting about this. So Bioconductor has a lot of packages, very specifically made to analyze biological data. And you can see right here, there is various packages, and they have their names here, who maintains them, and then little descriptions. And then over here, most importantly, is the rank, which is basically what package actually gets used the most, indicating that it's like actually useful. And then you can type in things like DNA, and it will give you things that have like DNA in it, data analysis, blah blah blah. So very useful. This is all the software, I believe, available. In this tab, we can like look at a little like more precise fields if you want to find something that's more specific to like something you're trying to do. Overall, there's a bunch of packages in here they're very designed like to handling biological data, both in its like formatted form, so like taking out a FASTA file and then like actually understanding and taking out the useful information that you want and then analyzing it. So very good. So today we're just gonna focus on one of these packages because I just want to show you how to like use Bioconductor. It's not dissimilar from normal packages, but I still want to get over it. Still want to like go through it, and as like more of an example. And the one I'm going to teach today is biostrings. So this is how you install biostrings, as it tells you right here. And this is how you figure out the version. When you click on, let me actually go back. Oop, well, not that. Once you go down here and let's just type in biostrings so I can find it. Like you find it, you click on it, takes you to this page. We're just on. I'm just gonna exit out of that, and then there's various useful pieces of information here. This, like right here, you click on this, and it gives you like information. Like usually, all the packages have like detailed documentation of like how things work, or like an example of like the R script. So if you want to learn more about a bioconductor um, package, you can just look inside this portion right here, and it tells you like, oh, you want to learn how to do multiple line. This is how you do it. This is an example. So these are very useful, and they're well documented and well maintained, and it's just good. It's just good for lab work and biological lab work. All right. So like I said before, we're going to look at strings. One shout out. This person, this is an older video. It's like 4 years old. Um, Originally helped me learn biostrings. Not everything of like my code is actually more up to date than his, but very useful to you and watch it. Um, like not everything he has there is still up to date, but it's still a useful video if you want to watch somebody else run this code. All right, what's now after all that? Let's get into it. Let's install biostrings. So Bioconductor Manager install, which is what they show you here. There you go. And then I'm gonna hit no again. And like any other package you want to use, you need to like you only need to install it once. Uh, Throughout your whole time you're using R, you only need to install a package once. You don't need to install it every single time you're going to use it. You just need to do it once. But you do, if you want to use it in a R instance, you need to call on it. You need to call it from your library into your instance. So you got to use the function library, and then you put biostrings in it. There you go. And then we're going to use ggplot to visualize the results from the biostrings. Okay. So for this, we're actually going to be using example data from this site here, from the NIH, and you can look through here and find various like genomic data. And specifically, we're going to be looking at, let's copy paste this. This E. coli data string. And the way that you get data from these is you go to their page. Like, let's just actually copy this and go from the start. Sorry about this. Go to here. Not looking like it should. Oh, because I think I might be in the wrong button. Well, it's just not working. Or I'm just using it wrong currently. So you get to the genome page, and then you can click on FTP, and it takes you here, and you want to get a FMPGZ file for this example. So this one right here. All right. Whoop. And to download the file onto your R instance, you can use the download file function, which is right here, which is you put the address of the data. So let's go over here, open a new tab. It's going to try to open, but you can just copy this and then put it into here. And then you put genome FNAGZ. And that, so this is the data source, and this is how it's actually going to download it as onto here. And after you have this data, which is just actually bring it up. We'll see if I can bring it up. Oh, there's. It's just a bunch of like genetic data about the various. Um, it's just genetic data from E. coli. That's all right. So this program. Sorry, this program BioStrings deals with genetic information. All right. So after we have that and downloaded, it should be downloaded as this name. And since it's that name now, we can use the biostrings function read DNA string set and put in that file. Let's run it. There you go. And let's actually look at it. And you can see here now the biostrings has this nice little formatted, color coded string of the nucleotides. And then you can use, there's various functions like alphabet frequency, which calculates the frequency of nucleotides in the genome. Let's use that. And it gives you a count of like A has this, C has this many, G has this many, T has this many. And then it has like the various possible symbols that that's used in files to indicate like different different instances in the sequence. And then you can then use you can use alphabet frequency again, but this time increase use as prob equals true, which indicates that like you'd want it to actually put everything not in numbers but as proportions. And let's look at that. And you can see now it's from these large numbers it's become proportions, which are all about 25%, which makes sense. And then if you want to remove the extra characters like these, like the MR, WS, so on and so forth, you can use the indexing of the data 1 through 4. So no rows, but 1 through 4 columns. It makes sense because this is the first column, second column, third column, fourth column. So this is basically saying, give me back the first 4 columns of this data. And there you go. And this is where the ggplot comes into play. We're going to create a bar plot of these frequencies. And there we go. A little bar plot, very basic one, but one nonetheless. And then you can call upon the frequencies individually. Very basic. And then, or you want to see what's the frequency, what's the CG percentage inside this DNA piece of DNA. You can run that. And, R is still mathematical, so you can I should probably explain this more. So it's gene frequency, which we have had before, inside, inside, and then you do the second value, and it's the second value because C is the second value, and then you do the third value because G is the third value. But you do these double brackets, so it only extracts like this portion. If you just did two, it would take like the whole. It'd actually take the whole second row. But if you did like indexing like this, it would take like this whole part, like the C and this, and the G and the probability, and it wouldn't be able to math because the data is wrong. So you need to put these brackets here to like make a subgroup of a subgroup within this to get that out. And then with that, you add them together because R can do basic math. I can do very complex math, but this is very basic math. And you get the percentage. There you go. All right. And then, for example, another thing that biostrings can do is actually they can locate certain sequences in the sequence. So you can essentially, we're going to say, hey, find the start codons. We're going to try to find the start codons in the sequence and like find the count of them and where they're at. And first off, we're going to assign the first type of start codon, the forward start codon, the P1, and the complement of that to P2. So there you go. Then we're going to use count pattern to count the number of patterns in the dataset. So you use the function count pattern, which is part of biostrings, and you put the first pattern and then the genome, which we assigned up here. So it's like all the genomic sequence again. Let's run that really quick. It's a whole sequence. And you're going to use once again, you use the method of like inner brackets, so the first brackets, and then the secondary brackets, because if you use only one bracket, like see all this, and you don't want that. You just only want to see like this part, like the bottom part, like the second column. So you need to use the secondary brackets to make a subgroup in a subgroup. All right, so let's then run this, and it's counting that there is 76,000 around of the start codon ATG in the sequence, as you can see there. And then we're going to do it for the secondary sequence. So once again, count pattern, the secondary sequence will be assigned to this, and then the whole genome again, and it gives us about 52,000. So you can also use match pattern to give it the position of the pattern. So this is a method of like being like, okay, this pattern is in this position inside the sequence, and I'm just going to show you how to do it. So use the function match pattern, the pattern again, and then the genome with the subgroup 1. So you only get the sequence again, and let's assign the first type of sequence to 1, and then do the second sequence to M2. And now let's look at it, and you can see here for the ATG, it finds it in this position. So start at 30 and at 32, and that makes sense. And with 3, and it keeps on going down, down, down. It creates this very long table, and it indicates like all the places it is, and like it's so much that like it doesn't show you all of it because it's too much, but it does. This is like good data. All right, let's now look at M2. The same type of thing. Failure start, end, and how long it is. And then it goes on and on. It shows you where this where the sequence is in the whole genetic sequence, nucleotide chain. And then we're going to actually turn these results I just showed you, this one and this one, into a data frame. And we talked about this before. The way that you can convert it to a data frame is as data frame, as data dot frame, and then M1, which we assigned here, which is like these tables at ranges. So you get only the ranges out, which is these parts. You don't get the width or this. You only get the ranges. We're doing that for the first set, and then we're going to do it for the second set. And let's look at the first set. And you can see it gives you a big old data frame. So you can clearly see all the data and it's very clear. And you could actually export this out as an Excel if you wanted to. Very useful. And then same thing here. And to my understanding, BioString is actually more flexible, so you can basically tell it like to be like less, to like look loosely for a sequence, so it doesn't have to match exactly. We're not going to talk about that right now. And then finally, we're going to actually use ggplot to make a figure that shows the density of each of like the occurrence of the sequence we're after, which is the start codon sequences. So I'm actually going to run that, and then once we have the figure, I can explain it better. So this is the density as the y, and then this is the start. So basically, it's where the sequence actually starts. Not the the data doesn't include the end. So the data is the matched. So this data frame we created, and then the x is the start, which is, let me scroll all the way up here, which is this part of the data frame. So it's the beginning of the pattern that we were looking for. And this saying, oh, this color and the kernel rectangle, it's going to use rectangles for this. And then the number is M46 for how many points it's willing to make. And then same thing for the second one. So you're creating two lines indicating the density of these two sequences you're interested in seeing. And then I'm in a theme to just give it a look. All right. And that's that. As a final thing, we can look at how to actually get read data from our computers, and it's just basically you just put in like the actual like length of the. Let me give me a second. You go to where it is, and then you copy as path, and then you paste in here like this, and then you use the read data string set directly. You don't have to do the download thing that we did for this other part. Yeah, you can see here. And the data. All right. That's it. The very quick introduction to bioconductors. It's a lot. I could cover a lot about this, but it's like too much and too specific, and I don't want to like. I just wanted to show the basics of how to use it, and like by showing a very straightforward example, because I don't want to like teach something that's not useful. Now that that's done, I'm actually going to do something else. We're going to, just give me a moment. All right. We're actually going to try to use an AI to write bioconductor code. I actually never tried this before, I actually want to see in our bioconductor. Yep. Uh, use biostrings to analyze genomic data. I just want to see what it does. Hmm. It's writing a lot of code. Um, I think it's literally just taking from like the examples that's in the file, like these, and just spitting it out. But it is doing it, so that's interesting. I don't know if it could. I don't know if it only would be able to do basic R functions, but it is able to delve into biostrings, which is pretty cool. So it's still probably, yeah, it's looking like it's getting this stuff correct. Can be used for bioconductor, but once again, learn how the tools work, and then use the automation system to help you make stuff, so you can like double check it. Uses fudgy messages. That's funny. Okay, cool. Anyhow, so that's Bioconductor. And it seems that the AIs can now use it too, which is interesting. And it is a wonderful depository for tools to analyze biological data. All right, now let's get to the homework. All right, so basically, the homework is pretty straightforward. It's just really just getting Bioconductor going on your instance of R. You go to this website, get the genomic data file from it, and then make a graph like this. Straightforward. Just you can basically just use the code that I already showed you and explore a bioconductor and try to find something that could be useful to you. Because there's some things that are very specific. It's like there's some bioconductor packages that are like literally just made to like run certain data types from like certain companies. And I'm actually going to cover that in the next class. But thank you for listening and learning with me. And I will see you at the next class.